Hello, and welcome to the Strategy webinar. Today we're going to explore abiotic stressors and the role that seed treatments could play. As many of you know, my name is Julie Deering. I'm the editor of Seed World, and today I have the pleasure of being your moderator. First, I'd like to thank our sponsors, BASF, Bayer, and Syngenta for their support. I also want to let you know that the webinar is being recorded and will be made available on seedworld.com following the event. As we go along, if you have any questions or comments, please share them in the chat box, and we'll do our best to address them. Today, our speakers are Kevin Bugg of Bayer and Molly Cadle-Davidson of Advanced Biological Marketing, also known as ABM, and she's the Chief Science Officer there. Today, we're going to discuss the most prominent abiotic stressors in the current landscape. We'll also take a look at seed treatments already on the market, in the future of these seed treatments, and the research that's being done and what Bayer and ABM are working on. After the presentations, we'll open it up for questions. As I already mentioned, if you have any questions as we go along, just type them into the chat box. So first, I'd like to introduce Kevin Bugg. Kevin is the manager of Bayer's Seed Growth Efficacy Research Team in Research Triangle Park, North Carolina. Kevin received his bachelor's in microbiology from Mississippi State University, completed his master's at North Carolina State University, and is currently working on his doctorate in plant pathology, also at North Carolina State University. He has been with Bayer for the past 11 years, and Kevin and his team have been valuable contributors in the development and launch of several seed treatment products, including Poncho Votivo and Ilevo. Kevin, the floor is yours.
Thank you, Julie. So today I'm talking about abiotic stress. Specifically, I'm going to be talking a lot about the interdependence of plant health and soil health. As we look at this graphic, I think many of you have heard the discussion about the rise in our population versus the amount of arable land, and how as the population rises, that leaves less arable land per capita. I think one of the largest problems facing our world today is the ability to feed everyone. The world's population is rapidly growing, and in order to secure our food supply, we need to produce more crops on the same amount of farmland. There's cultural dietary practices which are like the increased consumption of meat, which are certainly contributing to this issue. But in the same way, I think that there are cultural changes involving conservation and reducing waste that could help solve this issue. But is that in and of itself going to be enough? I think everyone recognizes that environmental challenges are rising, and that we not only need to sustain our current level of production, but based on this chart, we really need to increase our level of production. Much of the focus of agriculture has been, and I think will continue to be, combating biotic stress factors such as weeds or insects or disease. But a growing trend, which is not so much a shift of that focus, but a more all-inclusive approach, is an increased emphasis on abiotic stress. And I think that tools are being and have been developed to not only combat abiotic stress, but really to use the environment to our advantage. I think you mentioned in the opening that I work in seed treatments. Seed treatments are traditionally thought of kind of as stress managers. Historically, seed treatments have mainly focused on biotic stressors again, pests, fungal, bacterial diseases, nematodes. And I think you can certainly make the argument that plants under less biotic stress are better equipped and able to overcome abiotic stress factors. So this indirect effect has kind of always been there. But in addition to that, there are certainly examples of products or even classes of products that were developed with a biotic focus, which actually provide some plant health, plant growth benefits in the absence of that biotic pressure. A couple of examples I've listed here are strobilurian fungicides and neonicotinoid insecticides, which again are going to provide some plant health benefits outside of that main target. But again, it's really focusing, the focus of this seminar and kind of what we're really talking about today is abiotic stress is becoming more of a primary target and not kind of a secondary target. I believe that if you think about abiotic stress, it's sometimes considered maybe less complex, or at least something that maybe we're less equipped to mitigate. I mean, flooding, drought, extreme weather are inevitable concerns, and even less impactful environmental conditions. We have irrigation, we have fertigation, we have variety hybrid selection, and we do have some products which are focused on solving these, but I think that overall the arsenal of solutions and tools that we have is somewhat more limited when it comes to abiotic stress. This is where I believe that change is really starting to occur, and it's really a source of innovation right now in agriculture. There's a lot of new strategies being explored. There's a lot of new products being developed, products with a focus on and that specific target of mitigating abiotic stress. There are a lot of abiotic stress targets, such as drought stress or heat stress, but today the one that I'm going to focus on is creating healthier soil, which again I think in talking about this movement to look more at abiotic stress is certainly something that is of great interest right now in the industry. Certain environmental conditions are always going to remain out of our control, but it may be possible to help plants not only survive but thrive under certain conditions by manipulating different things in their environment. And one of those things that we can cause change in effect is nutrient levels in the surrounding soil. Along these lines, one strategy that's being explored is the use of microorganisms to expedite the breakdown of organic material and to ultimately make nutrients more available to the plant. In essence, I think that what we're doing is speeding up a natural process which provides more immediate benefits to the plants. The more common means of accomplishing this goal has always been to simply add more nutrients to the environment. But if we simply make the nutrients which are already present more available and make more efficient use of the nutrients which is already there, I think that that's an important strategy that we can use, again, as a way to control abiotic stress factors and help protect the plants. The map above, you may have seen something or something similar. I would imagine that a fair amount of our audience today are in the Midwest and in areas where soil health is not as much of a concern. They may be above average when it comes to nutrient levels and quote-unquote healthy soil. But again, the 2 points that I would bring to that is, back to the first slide, we were talking about we not only need to sustain our current production levels, but we need to improve them. And the second point I would make is that while you may have an abundance of nutrients available in your present in your soil, are those nutrients available to your plant, and are there things you can do to increase the availability of those nutrients and do that at critical times, ultimately helping preserve the yield potential of the plant. Every plant's going to face abiotic and biotic stresses, but healthier plants are better equipped to overcome stresses, and healthier plants in some ways begin with healthier soil. So soil health, there's a lot of things that we could talk about when we're talking about the topic of soil health, but I think one of the main factors is if we talk about the organic matter which is in the soil, because that's really the driver for plant growth. But again, all organic matter is not readily available to be utilized by the plant. Tillage practices, crop rotation schedules, soil types, and many other environmental conditions can affect the availability of the nutrients which are in your soil. And again, this is a naturally occurring process where this breaks down to become available for the plant, but what we can do to help that process along has become the focus of a lot of agricultural research. I think the bottom line is that expediting that process creates a healthier environment for the plant, and that we better enable that plant to reach its full potential in doing that. So plant health, again, plants are healthier, better able to come whatever stresses it faces, both abiotic and biotic. I think plants are more vulnerable to stress within the first 30 days after emergence, and that has contributed to the use and the value of seed treatments, which I mentioned is my area. We apply fungicides to plants to get them up out of the ground and provide early and sometimes long-lasting protection against diseases. Insecticides and nematicides along that line are also valuable tools to combat these potential biotic stresses leading to healthier plants. And it's along those same lines of reasoning that we're now exploring improving the health of the soil around the plant and these other abiotic stress factors that we can ultimately produce healthier plants better able to defend themselves against any and all stresses. And again, these abiotic and biotic stress factors are certainly interdependent, because if you protect against one, you're going to indirectly help provide protection against another. I think it's this concept of focusing more on the abiotic stress as a primary target, I think, is just another way that we can help improve the overall health of the plant. So along those lines, one of the directions that Bayer is going right now is a new product, Poncho Votivo 2.0. Right now we're planning for launch in 2018 for the 2019 growing season. So many of you are probably familiar with Poncho Votivo. Poncho has a long proven track record of providing protection from insects above and below the ground. And 6 years ago, we added a biological component known as Votivo, and this was to increase plant health as well as provide protection against plant pathogenic nematodes. And what we're bringing now in this next generation of Poncho Votivo is a second complementary bacteria with the focus of what we've been talking about here, improving soil health through increased microbial activity and therefore creating better nutrient availability. This concept that we've been discussing is not only being explored by Bayer, and it's being explored in several different ways across the industry. But what we hope to do with the Poncho Votivo 2.0 is to provide that technology, to provide that benefit through a seed treatment. And so there's not a separate application, but you get that technology, you get that benefit already on the seed and in the bag and ready to go. So that's really all I had today. Just wanted to kind of run through that briefly, and I will hand it back over to Julie.
All right, thank you, Kevin. Yeah, it's good to hone in on soil health and nutrient availability, and especially as we look at some of the global challenges the industry is trying to solve. Making use of what's already there is certainly a good strategy. So thank you for that, Kevin. Now we're going to switch gears a little bit, and we're going to hear from Molly. Molly's going to talk to us about drought and salt tolerance. Molly is the Chief Science Officer at ABM, and she works to enhance ABM's genomic strategies and to foster next generation product development. She is an expert in the field of genetics and is well versed in the application of genomics and next generation sequencing techniques for trait-based research and development. Prior to joining ABM, Molly was involved in government work with SRC and aided other government-funded programs with the Departments of Homeland Security, State, Defense, and Justice. Her work has resulted in 1 trade secret, 2 patents pending, and 1 patent application currently being prepared for the company. She holds a bachelor's in genetics from the University of California, as well as a master's in plant pathology from Washington State University, and a doctorate in plant breeding and genetics from Cornell University. Molly, the floor is yours.
Thank you very much, Julie, and thank you for everyone for tuning in. So today I'm going to switch gears a little bit. Kevin gave a very nice talk about the nutrient availability, and I want to switch gears to something we think about more commonly as abiotic stress, which is drought and salt tolerance. And some of our goals at ABM is across the board are to help the plants do more with less, because there's no question our world is changing, and so in terms of drought resistance or salt tolerance, we need increased water use efficiency in our crops and a drought tolerance in the field. And how those are just slightly different is if you're in an irrigated situation, you want to be able to produce a crop maybe using a little less water because it's becoming more and more scarce. And the image that I'm showing right here is just some of our field results. This was actually in a farmer's field in the drought in 2012 in Illinois. The plants on the left had no biological, and the plants on the right had our Sabrex product. And so this just shows the stark contrast between these two different treatments and where we want to go as a company with ABM. This figure shows why thinking about drought and salt tolerance are becoming more and more important, and as Kevin said, why abiotic stress resistances are coming to the fore as kind of primary goals rather than secondary. You've probably seen pictures like this before. The more red an area is, or even purple, the drier it is. And so the top image shows from 2000 to 2009 averages, and then the bottom image shows us going forward. And you can see in the bottom there's a lot more red to purple, and in fact, if you look at the picture as a whole, there is just a lot more extreme. There are a lot more extreme conditions. And what this means for us is that the condition of our arable land is changing to mean we're going to have to be farming with less water in most of the places where we farm. So how do we protect against this? After I made this slide and I passed it off to Julie, I realized I left off a really big way we can protect ourselves, and that's through plant breeding, whether it's conventional breeding or through some genetic engineering technologies or some more modern technologies. So that's an obvious point here. Through chemistry, we can create soil amendments that help the soil retain more water and kind of mitigate the salt increases that come along with drier environments. Cultural practices, improving soil organic matter and soil tilth, all these things enable the soil to hold more water. Even when there isn't water, what is there is retained and is usable by the plant. But where ABM is focusing in on is biological products, and we are wanting to leverage the plant microbe synergies. These synergies have been around throughout the evolution of plants and microbes, and this enables plants to better utilize available resources. Some common examples of this are listed right here, and probably the most notable one is the interaction of bradyrhizobia with soybeans, for example, or nodulation on legumes. This is a plant microbe interaction, a synergy that in which a microbe is fixing nitrogen to put it into a usable, a functional form for a plant. And without this interaction, we would have to apply much more nitrogen in different forms than we currently do. So this is one really classic example, and I've listed Trichoderma here as another, and this is one of our main organisms that we're working with at ABM. How these biologicals work with a plant, and specifically ABM strains, is they associate with the plant's rhizosphere, growing along with the plant root through the life of the plant. The other aspect that we really count on to enable these synergies is for the microbe to be endophytic, or an internal symbiont. And this allows a very close connection between the microbe and the plant to enable recognition of signals between the microbe and the plant. And so, because these are some traits in the microbes that we're counting on, this is a big reason why ABM's products are primarily seed treatments at this point. So we're putting our Trichoderma, and in the case of our legume products, our Rhizobia, bradyrhizobia, we're putting these right on the seed, so that as the seed germinates, the root, the emerging root, is actually growing through this zone of microbials that allow this rhizospheric and endophytic colonization to take place. Having colonized the plant root, then the microbe is established inside the plant, and this is one real benefit of working with an internal symbiont, which is that the microbe is actually protected from some of the soil environments. And that becomes really important if we're thinking about drought or salt, that it's an added layer of protection for that microbe to kind of buffer it against whatever the external environment is. Whoops. What this means then for the plant, or for this kind of synergistic organism, is that once we put the microbes on the seed and they colonize the plant roots, this allows the microbe and the plant to signal each other with metabolite chemical signals, then triggering signal cascades inside the plant that causes widespread gene upregulation in the plant. I should say that the microbes at this point are only on the plant roots. ABM strains, which are Trichoderma, Bacillus, Bradyrhizobium, Rhizobia, these are limited to the roots. And this gene expression changes, the upregulation that I'm talking about, is occurring in the above ground parts of the plant. What happens when this gene upregulation takes place is that whole systems in the plant are modified, changing the physiology of the plant. And the biggest effect that we see, or the one that probably is overarching with all these other effects, is enhanced photosynthetic efficiency. And the part that I'm talking about mostly today is resistance to abiotic stresses. This is enabled by this root colonization and gene expression changes by changing a very specific biochemical pathway inside the plant. Just to step back to explain this a little bit more, in times of stress, so in a drought situation or a high salt situation, plant cells produce reactive oxygen species. This is akin to in humans when a human is stressed physically, emotionally. However, humans produce free radicals. Reactive oxygen species and free radicals are really the same thing. And what humans can do is take a vitamin, an antioxidant, or drink a glass of red wine or eat some dark chocolate. All of these things have antioxidants. But plants can't do these things. They can't take vitamins, they can't run away, they can't drink wine. And so what plants have is an onboard system for regulating the amount of reactive oxygen species in their cells. And that's the pathway shown in the upper right of this slide, called the Redox cycling pathway. What this pathway does is take the reactive oxygen species and reduces them down to a harmless form. Without doing this, the reactive oxygen species are pinging around inside the cell, doing all kinds of oxidative damage, particularly to the photosynthetic machinery. And when this happens, photosynthesis is diminished or even destroyed inside that cell, And it's why we see in a drought-stressed plant a browning of the tissue. That's actually cellular death as resulting from photosynthesis death. So this whole pathway, it turns out, the redox cycling pathway, every single gene in that pathway is upregulated by root colonization of Trichoderma. So that means that the plant is able to essentially cycle reactive oxygen species faster. So as reactive oxygen species are created, they are also reduced, which means that there is no longer damage being done to the photosynthetic machinery, and Trichoderma colonized plants then are able to maintain their baseline photosynthesis levels even in times of stress. What this means, in particular, what I'm talking about today is drought stress and salt stress. So, so they have no loss of this baseline photosynthesis, which means that these plants can keep creating photosynthate, which leads to higher yields, more stress resistance because the plant is overall more healthy, larger roots, and more green biomass. And with more green biomass, that means more photosynthetic equipment, which means more a higher photosynthetic capacity over the baseline. So these are the mechanisms that we see for Trichoderma abiotic stress resistance. And just to sum up, our strains trigger the multiple desirable pathways. By triggering the increase in the redox cycling pathway, the plants are able to eliminate cell-damaging ROSs from their cells, and this protects the photosynthetic machinery, thus enabling the plant to keep photosynthesis up and running and produce more biomolecules that contribute to other kind of stress resistances, defenses, and conversion to yield. So we talk a lot at ABM about our ability to increase root growth, and this is particularly important when we're thinking about drought resistance, because the better developed root system you have, the more you're going to be able to take up what water is there, and the better you're going to be able to dig deep to reach the water, once there's less and less. And so what I showed you on the previous pages are how we can increase the plant biomass, and that biomass is both above ground and below ground. And the question has always been, how much do we increase that root biomass? The image on the left here shows what you usually see when you dig up some of our plants just with a shovel. On the left is untreated, and on the right has our Sabrex treatment on it. But we wanted to capture all of the fine roots as well and everything that's happening there below the soil. And so we set up some of the tubes, the root tubes that I showed in the previous picture, to try and assess this over time through the season. And the graph here is showing a control, which is the blue line, versus Sabrex root growth over the season. And then this slide shows kind of the final result after we harvested that. We saw, and what's shown in the graph here is the percent of the, what percent the root is of the total plant. So in the case of Sabrex, our Trichoderma product, that plant was 50% root. In the case of the control, it was closer to 45%. And so this reflects so the Sabrex was 110% of the control. And these are just some images of the roots. This was a pretty stunning picture, stunning result when we pulled this out of the ground, because as I said, when you dig it out with a shovel, you just get that root ball very close to the surface. But these plants are digging really deep. And I should say this was grown in a situation here in New York where we were actually in an extreme drought this year, and several counties in this local area here were declared a disaster. So this is a significant result here. So what this means then is we've established that we are increasing biomass. We're increasing it below grounds, and so what does this mean for our products going forward? This image shows kind of what we see in the above ground part of the plant. The left-hand image shows plants grown in the greenhouse with a water deficit. The plant on the right having been treated with trichoderma, and the plant on the left receiving no trichoderma. There's a really big difference in the effects here. And the panel on the right shows salt stress, a similar thing. The right-hand plant receiving trichoderma, and the plant on the left, no trichoderma. And so these plants, we now know, have a much larger root mass, more fine roots, and have been activated through changes in gene expression that are the result of trichoderma colonization. So I showed you this image before, and now you have a little bit more basis to understand why we're seeing the effects that we do. Again, this was from the 2012 drought. The plants on the right having been treated with trichoderma, these plants are still green because the photosynthetic machinery has been protected from reactive oxygen species that are generated through stress responses. The plants on the left did not have this buffering from trichoderma. We saw this again this year in our extreme drought here in New York. The plants on the left did not receive any trichoderma, and we're in the same field as the plants here on the right. And you can see for scale some of our happy workers there in the middle of the field. So in these changing times where we've got real changes in our world, in the condition of our arable land and the amount of arable land we have, we really need to think about agriculture on a lot of different levels. And so what we're doing here in our R&D department is we're, as you might have guessed from my introduction, looking at it at the genetic level, both from the microbe and the plant side. But in thinking about, in solving this question of how we deal with abiotic stress, we need to think about the phenotype on a lot of different levels, because this is a whole system. Plants don't operate in isolation. They do operate in the environment of the soil and whatever our atmosphere is doing, and whatever microbes are present on the soil and on the leaf surface. And so this is something that we've been implementing in our research here, looking at our new products and our pipeline from the perspective of the microbe, the very base level and its genome and what it's doing, all the way up to using remote sensing technologies to help us get both the overview of our trials, but also to tease out some of the things that we wouldn't normally be able to see using multi and hyperspectral imaging, and then layering all these data together. Because in the end, as I said, this is a complex system, and even biologicals can't make something out of nothing. I talked about how we protect photosynthesis by enabling the plant to reduce the reactive oxygen species that might otherwise damage its cells, and this kind of maintains or even increases the photosynthate available to a plant. But that's not all there is to the story. As Kevin discussed, there's a lot of nutrients that need to be made available or need to be available to the plant arising from the soil, and both of these things are what are going into the structural and functional parts of the plant and making these biomolecules that are important for us to be able to feed the world in the end. So just to sum up, our world is changing. I've said that many times. We need to be able to feed the world on a shrinking arable land supply, and this is going to become increasingly challenging as our environment changes. We have many tools that we can use to solve this problem, and here at ABM we're focusing on the biologicals and microbial realm, really helping the plant to help themselves. And approaches for this to solve what's coming ahead, in our future, in our agricultural future need to be systems-wide, that plants and microbes can't make something out of nothing. Inputs are still going to be required, and we need to think about this whole picture. And with that, I'll turn it back over to you, Julie.
All right. Thank you, Molly, very much. from our last webinar to this webinar, the focus that continues to be, and I think for good reason, on roots and the soil health, and how those play together for the betterment of the plant. And so at this time, we're going to go ahead and move into the question and answer session. If you do have any questions, please type those into the chat box. It looks like we've already got some queued up here, so I'll go ahead and pose those to both Kevin and Molly. So the first question that I have is, for the reasons you have mentioned with the importance of microbial health and microbial communities on abiotic stress tolerance with plants, would this show the benefits of metagenomics in commercial agricultural production? Molly, I'll let you take the first stab at that.
Sure. Yeah, I think that this is an area that's getting bigger and bigger, and in fact, I know that there are at least a couple of companies that you can send soil or root samples off to, and they'll analyze what's in your soil. I do think this is important because we, as I said, the environment is changing, and we will need to use all the weapons in our arsenal, so to speak, to be able to continue to produce crops in an efficient way. And this, using metagenomics as one of these tools, may be just the next step in precision ag.
And if I might just ask, could you give a very brief description of what metagenomics is for those of our viewers like me who maybe aren't quite that in-depth on their scientific backgrounds? Sure.
Metagenomics is looking at the genome sequences, or a part of the genome sequences from the microbial communities, well, from relevant microbial communities, and this means a lot of different things to a lot of people, but in terms of agriculture, often it means what's in the soil, the microbes that are in the soil. So really, you can take a root or a soil sample and isolate the DNA and sequence everything that you find in there. You can do this also from environmental samples that may include leaf tissue or anything you like, but for agriculture it's going to be root and leaf tissue for the most part. But sampling from the root and the soil, that's going to be the thing that we can actually address a little bit better. So if you learn what all the microbes are through advanced sequencing technologies, you learn what the microbes are in your soil, you might learn that, well, I wanted to plant soybeans in this field, but I have no Brady rhizobium here, and so I really need to inoculate, or something along those lines. That's just one example.
Thank you, that helps a lot. Another question from the same individual is, and I think this one's also directed toward you, Molly. What microbial communities or seed treatments would be beneficial for salt tolerance in soil? For example, any particular symbiotic bacterial colonies that can mitigate salt in soil to benefit celery crops?
Yeah, so I would say that, you know, I'm going to give a very limited answer there, because what we do at ABM is work with Trichoderma, which we have seen gives very good salt tolerance to plants that are colonized by Trichoderma. There are lots of microbes, including bacteria, actually probably mainly bacteria, that are like halophiles, so they like salt. And so I'm quite sure that there are companies or scientists out there who are looking at some of these types of organisms that may work with plants in order to mitigate salt in the soils. So I can't say much more specifically than I know our Trichoderma does have this very nice effect, and I also know that there are bacteria out there that work very well in soils. I mean, in salt-heavy soils.
Okay. Thank you. And then I'm going to pose this next question to Kevin. And one of my questions and one in the chat box kind of go together here. So there seems to be an increasing emphasis in the area of microbials and biologicals as seed treatment or microbial health. A, why should this matter to producers on the farm? And then B, from your perspective, what's driving this?
So, I mean, to answer first the question more that's on the screen, I think that industry's increased focus on microbes in general is just a recognition of the important roles that microbes do or could play in the health and the productivity of the plant. And then why that should matter to growers is it can obviously be an important piece, microbes into the system, an important piece of an overall strategy to help them increase production. To your specific question, I mean, I definitely agree that this emphasis is increasing. I see strategies based on reducing abiotic stresses. Even though I do see this increased emphasis, I see these strategies and these products currently right now is vastly underused. I think growers will always be somewhat at the mercy of the weather, but there are certain aspects on which they have more control. Planting dates, for example. Planting early is a risk that often leads to a reward. And I think that's what we really have to consider when we're developing these products and developing these solutions that address abiotic stress. Growers have tools such as fertigation and irrigation and different things, but these aren't always practical and in some cases even applicable at all solutions. But offering growers more and more simple and practical solutions that help mitigate abiotic stress factors, I think is really the key to getting these products out there and getting these strategies implementing, which again, back to the rewards, hopefully they'll be able to reap the rewards from. Julie, are you there?
Sorry about that. Yes, thank you, Kevin. Molly, do you have anything to add regarding what might be driving the increased focus on this area?
Well, I think some of it, I agree with Kevin completely how he talked about this, and I think another factor might be thinking about about human health and what we're learning about the microbiome, where humans have a lot of microbes in their gut and on their skin and everything, and having a healthy microbiome leads to a healthy person. If you don't have a healthy microbiome, then you can be sick, you can have heart disease or be obese or have a lot of other issues. This, I think, is kind of spilling over into agriculture, like understanding this kind of relationship between microorganisms and things like humans or plants. And so understanding this relationship, I think, is becoming kind of even closer to popular science now, kind of realizing that this is true. I think that's one aspect, is just kind of spillover from human health. The other thing is that microbes are the decomposers in the soil. They are what contribute to healthy soil, and without them you can't have healthy soil. And I think that this message is getting out there a lot more, and so growers are starting to think about these things more and more, and so it's being integrated into agriculture.
Great, thanks, Molly. It's interesting to think about how the human and the plant side interact, or how many similarities there are. The next question is, would using a hydrogen peroxide-based sanitation chemical cause more severe browning from increasing the kinetics of polyphenol oxidase?
if that one's for me, then I would say that it should. That would be my guess. I don't actually have practical knowledge on how this would affect it. However, hydrogen peroxide is a reactive oxygen species, and it is a molecule produced in plants in response to stress and leads to a hypersensitive cell death, so browning. And I would say the answer would be yes to that.
Thank you. And when comparing Sabrix to the control, was a seed treatment fungicide applied in addition to a biological?
Yes, all of our trials that we do, we apply our biological over the top of the standard chemical treatment. So our control is the standard chemical treatment alone, and our biological treatment is the standard chemical treatment plus our biological.
And then, Kevin, this next question I'll throw your way is, do we have any idea how prevalent microbial usage is in agriculture today? What percent of farmers are using it, or on what percent of the land and production?
So I don't have an exact number or percentage, but I would describe it as very prevalent and growing. Molly mentioned rhizobium, so that's something that's used a lot in soybean to increase nitrogen fixation. In the seed treatment world, we talked about Poncho Votivo, which has a biological component, and that is the #1 seed treatment product in the U.S., so it's on most acres of corn in the U.S. So, I think it's very prevalent, but I also think it is it's a growing part of the industry. As we recognize more and more the benefits of biologicals, I think there's more and more products being developed with a focus on biologicals.
Okay. And then one of the other questions here is asking about if these products are used and there's not an abiotic stress, are there downsides to using these products? And if so, what?
So, I think that, I'm sorry, was that for Molly? Go ahead, Molly.
One of you?
I think it could be for either of us. So could you repeat the question, because I was trying to scroll through and see what the question was. Or Kevin.
Sure, no, that's okay. The question is, if these products are used where there is not an abiotic stress, or where maybe the abiotic stress that is marketed is not necessarily present. And so, for example, let's say that it is a product that benefits when there's drought conditions. Say it's a wet year, there's no drought. Are there downsides to using that product? And if so, what? From
my perspective, the biologicals can be viewed a couple of different ways, and one of the ways is an insurance policy where you don't expect to see a benefit unless there is a stressor like a drought. But in practically speaking, in our data, we see, and our field trials across the country, we don't see the negative effect any more than what you expect from field trials, kind of normal biological variation. We see really nice benefits even in very high yielding years. So we had a trial just a couple of years ago where it was 300 bushel corn, and we saw a 30 bushel gain out of it. So no, I would say there's no inherent negative effect to applying a biological, even if there's no abiotic stress, and there can still be a very nice gain from doing so.
Thank you. And I've got, we're bumping up against our time here, so we've had some great questions come in. I've identified two more that I think are important and that most everybody can relate to or would be curious about. The first one has to do with the interaction of biologicals or microbes in addition to an active chemistry or that traditional chemistry, and how are companies, how are you making sure that they're compatible when applied to the seed and that they don't kind of counteract each other?
So I can take that one, Julie. So we develop, when we're developing a product, when the work that we have done with microbial products, that's one of the first considerations we have is compatibility. With the Poncho Votivo, for example, you've got bacillus, and it's actually in the same formulation as the insecticide, and within that formulation we have to have antimicrobials to prevent unwanted growth of microorganisms within that formulation. So we focus a lot on bacillus species because they form endospores, which are very stable. It's not only compatibility, but shelf life is something that we're very, as we look at these products and developing something that's going to be stable on the shelf and stable on the seed. So this is always, and as we move into looking at other like trichodermas or fungal isolates, as Molly is working with, you're going to be probably potentially putting this on a seed that also has a fungicide. So I think it is a very significant concern that you have to think about how these products are applied and if they're going to be compatible in existing systems, and also stability if you're talking about microbes and living organisms that you're going to be able to maintain viability of those.
And if I can add that one of the reasons why we use Trichoderma is that it is a very durable organism as well, and has a lot of actually fungicide resistant. That's a general property of our Trichoderma strains, is being fungicide resistant. And so in terms of traditional chemical compatibility, we have quite a large list of chemicals that we are compatible with, and so we really have no trouble applying our biologicals over a standard chemical treatment application. We don't currently sell anything that is in the can with chemistry like that. However, we have really nice compatibilities in application.
Great. Thank you, Kevin and Molly. And one of the last questions here that I'm going to ask has to do with glyphosate and its effect on beneficials. And I think as we see crops with the Roundup Ready trait or Roundup Ready to extend, how does or does glyphosate affect beneficials when they're on the roots or colonizing those roots? Does that matter?
So does that matter? Not really. I mean, we don't see that with Trichoderma. So with Trichoderma colonizing the plant roots, no, we don't really see a difference between Roundup Ready versus not plants. And so the glyphosate is not affecting the Trichoderma. There is an effect, with Bradyrhizobia, that some of these can be sensitive to glyphosate, but we do have a glyphosate-tolerant Bradyrhizobium in our mix that we apply, that we have in our Graphex and Excalibur products and Marauder products. So it depends on the organism whether or not there's an effect, but definitely not for Trichoderma, and at ABM we've tried to circumvent that with our Rhizobia products.
Great, thank you. And I have one last question here. How does climate change influence your work in this area? And how are you working now for conditions that could impact farmers in the next 10 to 20 years?
I'll jump in here. So climate change is a big deal, and the graph that or the images that I showed at the beginning of my talk with the how much rainfall any given area is going to get across the globe, that will change, and that is changing, so we really need to be thinking about these abiotic stress tolerances that we can embed in our products or provide management strategies for farmers. And as I said, it's a systems kind of thing, because no one solution is going to be the silver bullet. But what I can say is that with the biomass increases that we see as a result of colonization by our Trichoderma strains, getting both the above ground biomass and the below ground biomass, we're converting a lot of atmospheric CO2 to plant biomass that is going deeper than this the top tillable layer, and so there's a lot of carbon that's actually being converted from the air to the deep soil, where it is being sequestered for longer than just a single season. And so thinking about it this way, we're trying to enable growers to actually be part of the solution. Not only do we have the abiotic stress resistance that's embedded in our products, but we also have this added benefit of being able to pull carbon out of the air and put it deep in the soil, thereby improving the soil tilth and organic matter, as well as starting to bring down the atmospheric CO2. So this is all the direction that we're going at ABM.
And Julie, just to add to that, I would say, there's certainly documented trends in climate change, but in the end, weather is still somewhat cyclical, and growers are going to always have good years and bad years, and always face that uncertainty every spring. And I think, back to the other question of, how are these products going to, are they going to have a negative effect if a specific abiotic stress is not present? We develop products based on broadacre usage, and we're looking for, much like Molly was talking about, these benefits, overall benefits. And I think that, what we hope for is, in a lot of ways, seed treatments are insurance policies, because we put fungicides on because if there is significant disease pressure, we know they're going to provide a significant benefit. So with these abiotic products, we're developing these products to be able to better overcome these abiotic stresses, whether they actually occur or not. Because the reality is, with climate changes and with changes in the environment, we know that may be more intense down the road, but even in the here and now, there are going to be good years and there are going to be bad years. And can we help provide some protection and benefit to the growers?
Great. Thank you, Kevin and Molly. I still have a number of unanswered questions here, and so to those participants who we've not gotten to your questions, what I'll do is get those out to Molly and Kevin in an email format, and then try to get answers back to you here as soon as we can. And so we'll try to follow up with you by email so that you do get answers to your questions. At this time, I'd like to kind of start to close things up. I want to thank both of our speakers, Kevin and Molly, for sharing their expertise and giving up part of their day to participate in our webinar. I also want to thank our sponsors again, BASF, Bayer, and Syngenta, for their support. And also thank you to my Seed World team working behind the scenes to make sure that we have a successful event. Again, the presentation will be made available on seedworld.com following. And thank you so much for your attention, and I hope you have a fantastic day.